So hello everyone, and welcome to our third and final Lunchbox talk for Research Week 2022. Before we get started with today's event, I'd like to give a shout out to Meg Parlow in Communications for all of her hard work on this year's Research Week. Her name wasn't mentioned in the chat messages during our town hall this morning, but I want to be sure that everyone knows that these events would 100% not have happened without her help. So thank you to Meg. And moving into our land acknowledgement, we wish to acknowledge that the campuses of Algoma University are located on the traditional lands of the Anishinaabe, the Mississaugas of the Credit, and the Mushkegowuk Cree, as well as hereditary lands of the Métis Nation. We also wish to acknowledge that many of us are joining from Robinson Huron Treaty Territory, home of the Garden River and Batchewana First Nations, on sacred land set aside for education as envisioned by Chief Shingwak for our children and for those as yet unborn. As today's session is being held virtually, we would also like to acknowledge all the Indigenous nations across Turtle Island whose lands and territories we are each situated on. So a few housekeeping items. This session will be recorded and will be linked to the auresearchweek.com website under the Lunchbox Talk section, which you'll find at the top right-hand corner. Participant cameras and microphones are off today. Only hosts and panelists have camera and mic access. However, if you run into any audio issues during the webinar and can't hear, please try another method of listening in, such as computer audio or calling in by phone. And all of that information can be found in the link in your calendar if you're an AU member, or via the Zoom login information at the link found on the posters. Each presentation will be followed by a 5-minute Q&A from our audience. We will follow up on questions as time permits. Otherwise, with a follow-up email can be shared. Live captions will be included. And if you are a member of the media, please reach out via chat to set up an interview with our speakers or contact the Office of Research. So today we will start today's Lunchbox talk with Dr. Paulette Steeves, an Associate Professor of the Sociology Department and Canada Research Chair in Truth and Reconciliation. Her presentation is titled Reclaiming the Indigenous Paleolithic Period. Paulette, I'll let you start.
Thank you. Let me share my screen and get my PowerPoint up. Okay, if my PowerPoint doesn't move forward, please let me know. And I'm going to leave it in this format. Okay, so Tansi, thank you everyone for coming today. So my research focus questions archaeologists' denial of a human presence in the Americas prior to 11,200 years before present. I wanted to gain an understanding of how many archaeological sites in the Americas had actually been dated to older than 11,200 years. So North American archaeologists have consistently denied the presence of people in the Americas prior to 11 to 12,000 years ago. This has led to archaeological sites found to be dated older than 11 to 12,000 years being denied, destroyed, or ignored, and thus Indigenous peoples' links to the land and deep history being erased. So the question of early man in the Americas became virtually taboo in American archaeology, and no anthropologist or geologist or paleontologist who wanted a successful career would tempt the fate of ostracism by suggesting that they had discovered indications of a respectable antiquity for the Indian. Archaeologists are acutely aware of the possible implications of the earlier peopling of the Americas, which reflects on contemporary issues of identity, ancestry, and ownership of the past or present. As Orwell stated, who controls the past controls the present, who controls the present controls the past. So what I found in my research actually is that there are hundreds of archaeological sites in both North and South America that have been published in academic peer-reviewed journals that have been dated to earlier than 11 to 12,000 years ago. And there is evidence of mammalian migrations between the Western and Eastern hemispheres across millions of years. So there's always many forms of supporting evidence in every area of research. And when we're seeking answers to questions, it's important to consider all forms of evidence based on a holistic view of the past. So these are two maps that show some of those sites, not all of the sites that are in my book, but these are sites that date to older than 11 to 12,000 years before present in both North and South America. So what is some of the other evidence? So we know that mammals migrated between North America and land we know today as Asia, the Eastern and Western hemisphere, for millions of years. We know this because horses first arose in the Americas. To get to the rest of the world, they had to walk across a landmass. We also know that camels arose in the Americas, and they can't fly like those Canadian geese in the sky in this picture. And so they had to walk across a landmass. And these camels their remains were found and dated to 3.5 million years ago on Ellesmere Island. Another mammal who would probably like to follow the camels and horses and eat them was the saber-toothed cat. Saber-toothed cats also arose in America. The oldest basal species was found in Florida. So what are some of the other forms of evidence? Well, we need to listen to indigenous histories based in oral traditions. So oral histories have been credited with leading to the discovery of Pleistocene archaeological sites. One example is that of the Kisimwick site in Missouri, discussed by Ashley Montague. It's now known as the Mastodon State Historic Site. Montague wrote about an Osage oral tradition of about a battle between the great beasts that lived in the area and traveled to the area from the east via the Mississippi and Missouri River. The Osage held ceremonies at the site every year for thousands of years until they were forcefully relocated. So the Osage had told the story about the Battle of the Beasts. And what did archaeologists find in that very area later on? They found the remains of dozens and dozens of mastodons and stone tools. So here you have an archaeological site being substantiated by an oral tradition. Roger Echohawk argued that oral traditions which contain historical information can reach back over 40,000 years. And we know there are many oral traditions that tell of Pleistocene weather patterns, movements of sea ice, solar events, volcanic eruptions, and Pleistocene species and glacial lakes across thousands of years. So this is what North America looked like 11 to 12,000 years ago when archaeologists argued that people first arrived in the Americas. As you can see, there was hundreds and hundreds of miles of very thick ice. There was no food and sustenance. This was not a good time for mammals or humans to migrate. This is what North America looked like 2 million years ago. You have a subtropical forest across most of North America. You have a land connection between the Eastern and Western hemisphere. What's in the Eastern hemisphere? Glad you asked. Lots of archaeology sites that date from 23,000 to over 2 million years. We know that people had left Africa, early hominids and later modern humans, and reached Northern Asia and settled the area as early as 2 million years ago to 24,000 years ago. So we're supposed to believe that even there was a land connection and forests and mammals coming and going, that humans had reached Northern Asia and decided they wouldn't go any further. That doesn't make any sense. This is a map of the North American sites, and you can see there's not very many in Canada. That's not because the sites were not there, maybe. It's because we don't disturb the land in these northern areas. A lot of archaeological sites are found when people are building roads or construction, and oh, there's an archaeological site, and then they get recorded and published, and then onto a database and onto my map. So there's lots of areas in North America that may have intact historical resources. This is one of the sites that's in my database. This is a Lacina mammoth site. I worked on this site post-excavation. It was excavated for 11 years. It was a field school. A lot of archaeologists worked here. Now that the site is not being excavated, there are still intact areas where mammoth and mastodon bone erode out of the cliff face And archaeologists go back every year and collect those and record that data. So I was doing that for my PhD dissertation. And this is in Southwest Nebraska. So when people were at this site, it was about 500 miles south of the edge of the glacier. And this site dates to over 22,000 years before present. So the archaeological sites I discussed today are not just archaeology sites. They're homelands to Indigenous people, places where our ancestors have left us their stories in the earth, telling us of their time in life here. When our homelands are denied and we are cut off from the very root of our existence, this is an ongoing and violent act of aggression, and it is completely unnecessary. Our work as archaeologists is to understand and record the human past, not to deny it based on bias and racism. The existence of hundreds of ancestral sites in the Pleistocene creates a dialogue from which Indigenous people can challenge erasures of their histories. It foregrounds their Indigenous identities and their links to lands, and it empowers them in seeking justice. So my research has shown that the denial of earlier than 12,000 year before present archaeological sites in the Americas is political, and it remains an ongoing practice of a colonial mindset that has worked to erase Indigenous history, diversity, and humanity. The acknowledgement of Indigenous peoples to the land in deep time is very important discussion that when woven through education and general discussions works to combat racism and discrimination and works to inform federal and state laws, policies, and land claims. Documenting stories on the land reclaims a history denied and hidden by colonizing nations and archaeologists embedded in state policies of assimilation and erasure. And that's and this is a picture on the bottom of my book that was released in July, The Indigenous Paleolithic of the Western Hemisphere. In this book, I reclaim over 100,000 years of Indigenous history in the Americas through archaeological sites and oral traditions, environmental studies, genetics, and linguistics. So sorry, that was a very quick recap of my research. And if anyone has any questions, I'd be happy to answer them.
Paulette, we do have one question in the chat, which is where would we be able to purchase your book? Where would you prefer that we purchase it?
Well, I don't have a preference. They do have some here in the bookstore, or you can order it online directly from the publisher. There is a audible version that's coming out by this summer. And there in 2023, there will be a paperback version.
That's great. So I'll let someone else, if anyone else has any questions for Paulette before we move on to the next speaker. I don't see anything in the Q&A. If no, Elaine has her hand raised. Laura's saying thank you for sharing today, Paulette. Important work. And Elaine, I don't know if you can type your question instead of.
Jody had a question. Could you provide more detail on all the elements, especially for long-term goals and how to achieve them?
No, actually, it was.
What is that?
What exciting projects are you working on these days?
Oh my goodness. Yeah. So doing a deeper dive into my research, I realized, how absurd it is. Vine Deloria Jr. called, the archaeologist's story of Indigenous history is absurd and didn't make sense. And he was right. A lot of it's Clovis first has been disproven. But I, really began to think about that question. Wait a minute. Early hominids were in Northern Asia 2 million years ago. And we're supposed to believe they just sat there that whole time and didn't go another, 4,000 or 500 kilometers to cross into the Americas, when mammals were clearly going back and forth across the Americas. So when I look at these archaeological sites that date to over 100,000 years and 200,000 years in the Americas, I can see that it fits with the global understanding of human history. And if you've noticed around the world in the last 10 years, a lot of the story about human evolution has changed because science has been very gracious in giving us ways to understand and clearly define deeper human history. And so it's so exciting to think that there are these amazing new technologies where we can pull human and mammalian DNA out of soils, right? And so I'm really now looking at getting a deeper understanding of the paleo environments and the plant and animal species in both North America and Northern Asia from 2 million years ago to present, and looking at where are the older sites in the Americas and planning future field work. Because I think it's really important to Indigenous people to reclaim these histories, and it's really important to the general population to understand that these histories that are claimed to be solid scientific data by white archaeologists are not supported by any data, they're hypothesis. And yet here we have the data that shows a whole different story, including the most recent site at White Sands, which they dated human footprints to 23,000 years ago. If you have people in New Mexico 23,000 years ago, and you're an archaeologist, you should be looking around where were their habitation sites, because they didn't just fall out of a plane and land there and walk there once. They walked there from somewhere else in the continent, and everywhere they walked and went, they lived. Those would be archaeology sites. So that's really exciting for me that more sites are being published, and the science and technology is there now to further support this research.
Thank you. It sounds fun.
Thank you, Jody. And we do have a question in the Q&A from Ozma. So she's saying, Paulette, thank you so much for sharing your research. I really enjoyed the CBC interview that you did. My question is, does the research impact on your understanding of the area around Lake Superior? And has there been any archaeological work done in this region specifically?
Yes, there has. And actually, an archaeologist named Bell was fired from the Museum of Man in Ottawa a few decades ago for claiming that a site in Manitoula Island dated to over 20,000 years. And there are other reports of older tools in this site. And to really study this area, which I'm just starting more to do, we need to understand what we call the glacial lakes. So what we see as Lake Superior today was actually a much bigger lake. There was a lot more water in some of these older Pleistocene lakes. There has been a lot of work done by archaeologists previously on understanding the high stands of Pleistocene lakes. And so I'm just beginning to work on a map to understand the sites in Ontario where we might find these older archaeological sites, and where people we know usually lived on a really large, good body of fresh water. And so knowing these ancient glacial lakes and their high stands would be an amazing place to do some core testing and testing of soils to look for possible cultural sites. So yeah, Ontario and this area is on my radar. Archaeologists have done a lot of work. The Royal Ontario Museum does have some collections, and it's really fun. It's like doing detective work and using the clues that other people have left to create new understandings of early humans.
Great. Thank you, Paulette, for answering those. I don't see anything else. Oh, Ozma saying, very interesting. I'm really happy that it's on your radar. Feel so fortunate to have you as part of our research community at Algoma. So yes, I would also second that. Wonderful. So I'm going to move on to the next speaker, if that's okay, Paulette. I don't see anything else coming up in the chat or the Q&A, but if there are any additional questions for Paulette, feel free to reach out to her or reach out to the Office of Research and we can connect you. And so next up, we have Dr. William Bill Dew, who is an assistant professor in the biology department. The presentation he will share with us today is titled Sense and Sensibility, How Chemicals Affect Chemosensation in Aquatic Organisms. So Bill, I will let you start sharing your screen if you have something to share. Perfect.
I do. Okay, hopefully that's coming across okay. Wait, hit the share button, Bill. Okay, great. All right. So thanks for that. As mentioned, my name is Bill Dew. I'm a prof of environmental science in the Department of Biology. And yes, I've made a very cheesy title slide. It's one of my favorite parts about my research are the puns that I can make based on my research. So my research is focused on animals in aquatic systems, so aquatic animals, and I'm interested in how they can perceive their environment through chemical detection. And primarily, I'm interested in their sense of smell or olfaction. And to take it a step further, I'm also interested in how contamination in these bodies of waters can impair this ability. It's a sublethal effect, so it doesn't kill the animal, but we can get effects at concentrations, below what is deemed safe by agencies. So you may not realize it, but fish do have noses. On the top left is a lovely little trout. Now you can't really see it that well. There's a little spit of tissue in front of the eye, which is basically the nose. You can see it really well in the bottom left-hand sturgeon picture. In front of the nose, you can see these two openings. One is called the incurrent nares, one's the excurrent nares, basically nostrils. The water goes in the front and out the back. The noses aren't connected to the respiratory system like our noses are. They can't go and smell. The water moves across their nose, and inside, underneath that flap of skin, is an olfactory system. There's a little cup which holds what's known as the olfactory epithelium, and I'll show you a little bit more about that in a bit. On the right-hand side, you can see a boxfish, which has little openings for its olfactory system as well. And you may have noticed that one of these things are not like the others. The top right is a crayfish. Now, crayfish don't have a nose, but they do have sensory organs on their face, so sensilla and setae and other things that they can use to perceive their environment. So let's just fly up the nose of the fish for a moment, just to get an idea of what's going on. And so this is the top-down view of a fish. It's been dissected out to show you the entirety of the olfactory system. So on the this part here is known as the olfactory epithelium. It lines the olfactory opening. The olfactory pit, if you will. It contains flaps of tissue known as lamellae. And within those lamellae, we have a whole bunch of different types of cells. One of the different types of cells are your olfactory sensory neurons. And this is neural tissue that is present in the system. It's what is bound by cues in the environment, and it sends signals to the brain. So this is in constant contact with the external environment, and the rest of the system isn't. So when an odor comes along and it floats into the lamellae and it binds to a cell, that cell will react, will send a signal down the olfactory neuron to the bulb, to that long tract, and then to the brain. And once it hits the brain, we'll have some sort of response. So you may ask yourself, well, what can a fish smell? Well, all sorts of things. Fish will use their sense of smell to find food, to detect predators, to detect predation events. If their buddies are eaten by a fish, it actually releases cues that it can detect, and they freak out. I mean, their buddy's just been eaten. They can also follow migratory routes. If you think of salmon spawning, coming back to natal streams, that's all olfactory based. They can also recognize kin. They use it for mate selection, so pheromones, and they can also use it to recognize spawning beds. And so there's a lot of behaviors. There's a number of different behaviors that are really important to fish. Now, if you want to go deeper into the cell, and don't be scared by the signal transduction pathway, we have the surface of the membrane, and you can see the little rectangle, which is the R. That's the receptor. So it's sticking out into the environment. It could be bound by an odor. It causes, when it gets bound, it causes a change in the shape of the receptor, and it causes all this pathway stuff to happen. And so there's a lot of really interesting steps within the pathway. So a lot of targets for contamination to interfere with. Eventually, it'll open up a channel and it'll allow calcium to rush in. And when calcium rushes in, the nerve fires. Okay, so we have this really nice complex pathway. Different types of olfactory sensory neurons will have different types of pathway, but in the end, they'll all respond to odors. And I can measure this response by sticking a probe up the nose of a fish. This is a fish. It's anesthetized. It's undergoing what's known as an electroolfactogram, or EOG. I put a probe between those flaps of tissue in the nose, those lamellae that I previously mentioned, and I have a tube bringing in a scent. then I look to the computer, and the computer output is on the right-hand side. We have a nice little, I'm measuring the charge around the neurons. I add something in, and then there's a response. All those cations rush in. And so I can see how big of a response, how well that fish can smell whatever it is I'm giving to it. Now, if I give a contamination, if I add copper or I add, an organic chemical, and I see a reduction in that response, then I can say, well, now this fish cannot smell as well. And I get this olfactory impairment at the level of the tissue. The other way is much easier to envision, and that's behavioral mazes. Now, this video is a video of a behavioral maze with a crayfish that an old student of mine did. They put the crayfish into an area to acclimate. In one end of the maze, he put a smell of food. In the other end of the maze, he put nothing, or a blank. He let the animal walk around the maze, and what'll happen is if the animal can smell, it'll spend more time in the arm with the cue. Now, if it's a food cue. Now, if it's an avoidance cue, it'll spend less time in. So we can measure the amount of time it spends in the arm or in the other arm, and we can tell whether or not the animal's attracted to the cue, whether it's avoiding to the cue, or whether there's no response. And so we can compare a clean animal to a contaminated animal. Now, over the years, I've done a number of different contaminants on fish, and there seems to be three main contamination effects, so patterns of contamination. One of them is we can give a contaminant and measure the olfactory response, and there's an impairment. But over time, even with the contamination present, it'll recover, and it'll be able to respond as if it was in a clean environment. So we call that acclimation or acclimatization. Another type of impairment we see is an impairment that lasts as long as the exposure lasts, but as soon as we remove it, the animal's fine, which is great for the animal, and it's great for contaminated environments because it means when you clean a contaminated environment, these animals will recover very quickly. The third type, and this is the most severe type of contaminant effect, is that after you remove the contaminant, you still have impairment. And at this level, you're talking about damage to the cells and damage to the tissue itself. I'm going to go through a bit of my research showing a couple of these different types of contaminant effects. So here's the first one. Lots and lots of bars on this graph, but basically there's what I did was I exposed fathead minnows to increasing concentrations of copper for increasing durations. And if you look at each individual group of four, each one is at a different concentration, and each different color on the bar is an increasing time frame. So if you just look at the four bars on the far left, after 1 hour exposure at this very low concentration, there's a big impairment of the response of the tissue to the cue. But over time, as you go up to 96 hours, it's fine. So this animal is able to deal with the contaminants at the level of the olfactory sensory neurons, and it doesn't really matter that is there after 96 hours. Now, if you're really worried about what's happening in 1 to 24 hours, then yes, this is a big effect. But if you're worried about long-term effects, it's not so bad. Now, if you go up to the third and the fourth set of bars, you can see that we have that idea of recovery, but we have an impairment that doesn't go away. And so this is kind of like the second type of contaminative effect, where you've got an impairment, you take it away, and it should recover. But as long as the contaminant's there, we're having this impairment. Now, another example of this is work that's just been collected by Andrew Arndt, who's a thesis student of mine. In fact, he's out in the hallway right now presenting his poster for our poster session here in biology. He did an experiment where he used crayfish and attraction to a food cue, and then he contaminated some of the fish with 5 micrograms per liter of copper. And I say that concentration because that is what is deemed to be safe in aquatic systems in Ontario. Now we can see in the control animals, they have movement, and we can see they spend about 50 to 60% more time within the food cue arm than the non-food cue arm. But when you add copper, there's no response. So we've lost the ability to smell. Now, he then took these animals, he put them in clean water for 24 hours, and there was complete 100% recovery, which is quite exciting. So it's saying that as long as the contamination is there, we're having effect, but you remove that contamination, we're good. So if you have a lake that's contaminated, by working to reduce the contamination in that lake, you can actually improve the health of the species by improving their ability to smell. Now, what's really exciting is this can be on a really long time scale too. This is a graph just showing work I did with a colleague, Ali Azizi Shirazi, in the Sudbury region. I'm sure you know Sudbury is very contaminated. We went to a variety of lakes. Hannah Lake and Ramsey Lake are both within Sudbury. They're within the zone of impact, so they're fairly contaminated. Hannah Lake is much more contaminated than Ramsey Lake. And so what we did is we took the fish and we put them into clean water. We put them into water from Geneva Lake, which is outside the area of impact. And we saw that when you put them into clean water, they smelled a lot better. They were able to smell a lot better. I don't know if they smelled a lot better, but they were able to perceive cues to a much higher degree, which is really exciting. So here we've got Sudbury, which has been contaminated for 100 years, 100 plus years. These lakes have been under this constant pressure of contamination. But if you put the fish into a clean environment, everything looks good. It's a little more nuanced than that. Ali went back a couple years later and he did this experiment where he looked at behavioral responses. And so on the column on the left, you have the Ramsey Lake fish in native lake water and in Geneva Lake water. And you see that when you put the fish in Geneva Lake water, they do respond. This is response to a predator cue, so they spend less time in the stimulus arm. However, the fish from Hannah Lake, they didn't recover. So this graph is kind of an example of contaminant effect 2 and 3, where you have an effect, but as soon as you remove contaminant, it's fine. And where you have an effect, but when you remove contaminant, it's not fine. There's a lot of examples in the literature of the last one, of number 3, which is that you put a high concentration of copper in, you kill the olfactory sensory neurons, and it can take a month or 2 for recovery. So you have to be in water for a month or 2. So we can have some really long-term effects. There's also questions about whether or not you're affecting long-term behavioral responses. You're basically causing evolutionary changes, but that has yet to be measured. Now, in addition to that, I wanted to mention a couple of the other things we've done in the lab. So at this point, I've just looked at what happens when you contaminate an animal with copper. We've used a lot of other different contaminants as well. And this one was just recently published. This is clothianidin. Clothianidin is a neonicotinoid pesticide. Basically, if you've heard about colony collapse disorder, one of the things that's been tied to is the use of neonicotinoid pesticides. These pesticides are really good at killing insects, and they're really good at staying in the environment. And I was interested in, okay, well, what happens when I wash off agricultural sites and into water? So my student Lee did a number of experiments with this. He found that they can't smell. I was quite surprised. I was actually thinking that they would be able to move less, because that's how this pesticide works. It causes the animal not to be able to move, and they die from starvation or dehydration. They were able to move just fine, but they were not able to smell, which makes sense because the target is actually involved in neurochemistry, especially in the olfactory bulb. We were also able to, I'm not going to show you the data, but we were also able to connect this to dominance hierarchy. When two male crayfish come together and fight, they'll fight. At the same time, they'll pee in each other's faces. They'll share urine cues, and one will win, one will lose. If the animals come back together, they do the same thing, but they'll recognize that they fought, and the one that's won said, "I won," and the one that's lost said, "I am so sorry," and they will back off, and so it'll be a very quick interaction. If they come together again and they can't smell the urine cues, they have no idea that they fought. They can't recognize each other with vision or touch, and so they have a full-on fight. And so the dominance hierarchy formation, there's issues with it when they're unable to smell. And the last thing I wanted to show you was some work I did with a master's student by the name of Kathy Sakamoto. We went to a lake sturgeon hatchery in Onaway, Michigan, and we exposed sturgeon to lampreycide, or TFM, which is used to treat for lampreys. We have a lamprey issue in the Great Lakes, and we the DFO does a lot of spraying of streams to kill off the amicetes, and so we can reduce the lamprey population. And so she was interested in what effect this was having on lake sturgeon. And so we use young of the year lake sturgeon, and on the left we have the physiology. So this is EOG. If you're exposed to TFM, they can't smell as well. In the middle, we have behavior. It was to a food cue. So there was movement towards the food cue in clean animals, but in contaminated, they had no effect. And then the last one was really kind of interesting. It was a feeding response. And so She measured the weight of the food that was fed to the animal and then what was remaining, and found that the exposed animals were not eating nearly as much food. So it's a really interesting connection between a physiological level with a behavioral level with a meaningful behavioral response. Currently, we don't know what happens if we remove lampricide. It's actually something I've been following up on, trying to get funded to see if we can figure out that, is this a long-term effect, or is this an effect that ends as soon as the treatment ends? All right, so I hope you've enjoyed going up the nose of the fish. And if you have any questions, I'd be happy to answer them as best I can.
Feel free to enter questions into the Q&A, or for those who are panelists, you can also write in the chat. I'll just give it a moment, Bill, because people might be typing.
Sure.
Oh, yeah, here we go. So Donna is saying, I did not know about fish noses, neither did I actually. So no question, but thanks for the interesting presentation. I would concur. I had no idea that fish they had noses, but I'm an arts major, so I have an excuse. And Narosha is also saying, thanks, Bill. She's wondering if you have explored species effects.
Yeah, we have looked at different species of fish. We see similar patterns, but we had designed a project to look at about a dozen different species. It didn't come to fruition, but the little bits of data we have show that the perch and fathead minnow seem to be similarly affected by copper. Yeah, so there's a bit of evidence to this, but there isn't much in the literature.
Okay, and Elaine's asked a question as well. She's saying, if we don't know the residual effects of lampricide in the context you described, why is it used so widely, and what potential risks are known and accounted for in this practice?
Ooh, it's used so widely because before they started using lampricide, there was a collapse of the fisheries in the States, and because we want to protect lake trout and we want to harvest lake trout and eat it, so they do the risk assessment. Now, the good thing about what's going on is there are constant measurements of non-target mortality when they do lampricides. And I actually have a student who works at the DFO who's trying to see, is going through that data to see what's up with that and to see if we can connect it to anything specific. But it's the economic importance of it. Now, the idea is that TFM breaks down really quickly in sunlight and it can wash out of a system. These systems are all rivers, and so you dump it in the river and it flows to the lake. So it should flush out of the system in short order, and it should break down fairly quickly based on the sun. But I'm not an expert on lampricide, so I couldn't give you any more details than that.
Okay, and Narosha has a general question. Do you know if there is any correlative effects of lampricide to human behavior or physiology, as humans are known to consume fish?
Well, lampricide is nasty. It like, if you were to, they have protective gear when they put it on at the concentrations they use, and I know there are some warnings, like when they do the Gully River, they'll tell people who are in well water near the Gully River to not drink well water for a certain number of days because they're worried about it seeping in. So it can have an immediate effect. The residual effects, I've not seen any data that says the TFM stays within the tissues, especially the muscles. But that doesn't mean that's not an issue. It's just it means it may not have been studied yet. Yes, always Narosha, always a collaboration. Yes.
Okay, well, thank you so much, Bill. I'm going to move on to the next presenter, given that we're running out of time here. But thank you so much. This was super interesting. And I'm sure that if anyone has any additional questions for you, that they'll reach out. I may actually, because this is super interesting.
Perfect. Thank you.
Great. Thank you so much. And so next up, we have Dr. Jody Rebek, an associate professor in the School of Business and Economics. Her presentation today is titled AwakenU Exploring Courageous and Conscious Leader Development Through Contemplative Training. And Jody is joined today by student Haley Casey. So I'll let them share their screen and get started.
Okay, so welcome everyone. I hope you can see me and see the slideshow there. I just put on the music because the research I do is looking at the impact of unseen messages. So nothing real physical for me to present here today. So I'm going to start just by giving you an overview of the research approach that I use, as well as looking at defining leaders versus leadership. And then I'll go into the context of some past undergraduate leadership development research I've done, or leader development research, and then into the character, which really looks at the AwakenU research that we're analyzing the data for now. And Haley's with me, and she's going to present just a few of the preliminary findings, just to give you an idea of some components of the research, what we're finding. So AwakenU is a project that I'm collaborating with colleagues Dr. Murigan and Dr. Rouleau. Haley has been the research assistant, and Hannah Kaiko and Jordan Barone are also assisting with this research. Hannah's getting her PhD right now, and Jordan's a current student. And in the past, Cameron Myhel was also a research assistant for this project. So this is a process I use for all of the research that I conduct in different ways. And I start out usually with the water section, really looking to design and affirm questions with people that I'm working with. So in the research I do with undergraduate students, they give me feedback about what they would like to do about the research questions, as well as the different interventions I've used with them as well. And then collect the data. And then I move to the sunlight, which is member checks, analyzing and compiling the different data that we have, basically to offer and shed light on the research that we're doing, which to me leads to growth, which I see as all parts of the research phases. I'm always taking research notes as I go through it, both for myself personally, as well as the content I'm exploring. And this is where I'm looking generally, once I've finished analyzing the data collaboratively with others, is to figure out what the action plans are, what growth is needed, what do we need to change, and also to encourage learning as well as follow-up and improvement. And then the seed phase is kind of like the diagnosis, where you just look at reformulating ideas and movement forward, so that you can engage others and start developing or improving that research as you go along. So I thought I'd share the research that led to AwakenU, which is the undergraduate leader research I've done in the past. But I wanted to ask a question of the panelists, because there is still not a strong clarity within the research around the definition of leadership. There's some consensus on some of the context of that definition, but if you could just put in the chat how you define leadership.
I'm not sure that everyone would have access to chat, actually, Jody.
Oh, okay.
Just
Okay,
because
so
this is
just
a
think about
webinar.
it. Yes,
Yeah.
okay, so just think about it then. Think about how you define leadership. I mean, even just answering the question for yourself will be interesting, I think. Thanks, Sarah. So for me, this whole question, like what is leadership, led me to really focus on leader development. And for me, there's a difference between leadership development and leader development. And there's other scholars in the research that also agree. Day is one such scholar that really differentiated for me the difference. So leadership is a process. You know, when you talk about effective communicators and problem solvers and people who are motivating, that's leadership. But leader is the actual character. of the person who is communicating. So I really wanted to focus on leader development because there hasn't been a huge amount of attention on that character development trait in the business undergraduate literature. And then I was looking at, I analyzed about 12 different theories, the more contemporary theories and tried, tested, and true theories of leadership, which found that self-awareness for all those different theories was foundational. So that's what I want to focus on with my research and see if engaging students in self-awareness development could lead to insight. So this is Brene Brown. She says, leader is anyone who holds her or himself accountable for finding potential in people and processes. The term leader has nothing to do with position, status, or number of direct reports. So in this, I want to reframe, what traditional leadership has been seen as. So for me, a leader is someone who's kind of makes that roadmap and plants the direction forward to getting to that common goal. It's people who stand up for justice. It's people who share their voice and have the courage to speak how they truly think and feel in a meeting in a kind way. So kindness is a really big part of leadership. People who share, even at a young age, like Autumn Pelche, who take a stand for something they think is important, not because it's easy, but it takes a lot of courage for young people like her and this young advocate for the environment. So for me, those are leaders that kind of take a stand, share their voice, and lead other followers towards a direction. So what does it mean to dare greatly? This is Brene Brown's work where she looks at leadership from a point of not armored, where you're focused on ego and you're protective, but you're actually brave and courageous, and that means you're vulnerable. So I'll get more into that in a minute, but basically it's not a position, right? We do have people who are in higher up positions of leadership. So I'm not focused on that because that naturally has some followership attached to it. There is leaderships of countries, of organizations that obviously other people follow. But I wanted to focus on people of influence. How do we nurture a leader in a way that they're able to have influence and engage others in good work? So daring leadership here is what Brene Brown shares is rumbling with vulnerability, living your values rather than simply professing them, braving trust. So being the first one to share or to connect or reach out to others. And then learning to rise, right? Like it's not easy to lead and be vulnerable, and you might fall, but it's having that courage to get back up again, to learn from your mistakes and see that real beauty of learning. These are my two boys, and they're, to me, what I'm hoping to nurture in my family, that sense of I have a voice and I have something important to share as well, especially in the interest of kindness. By the way, someone else went first in that hole. So context is where I want to just share some of the learnings from past research. So the research I did with undergraduate students focused the social change model of leadership, and it has 3 components. And this is why I love it, because it starts out with conscious. How do we look at, who we are? The second part is congruent. And the third is commitment. So what I had that I didn't think was significant with the research is I had a meditative intervention for students. They decided on the intervention. And we had this 3-segment reflection that they would do after each meditation. We had 2 classes a week. We'd start with a 5-minute meditation. Like I said, they designed this. And I had them do a reflection on their thinking, their being, and doing before they meditated, during, and after. And so what I found there is that there was a real integration between this leader development and the meditation experience. The meditation allowed them to become a little more cognizant of what their thoughts were, what their feelings were, and how their behavior either aligned or didn't align with their values and what was important to. So it's a practice here where a lot of people felt it relaxed them. It was a process that helped them to transform their mindset around who they were. And it gave them this focus and a more compassionate attentiveness to themselves. One interesting thing for me was that a lot of them said they were more open to feedback from others. So that was for me, I love feedback. So it was a real shock for me to hear that they really didn't like feedback before, that they thought it was kind of like a hit against their character. Whereas through this process, after only 8 weeks, it wasn't long, that they had felt that they were more open. So this is something that I had reflected on after this study. We are multidimensional beings, but in education, we're focused on a one-dimensional learning and leading. So transformation to explore the other dimensions is key to healthy living, leading, laughing, and learning. So I'm going to go into the AwakenU study that really focuses on character. And I was a bit inspired to engage on this because of the COVID-19 pandemic and the fact that so many students were struggling with mental health. And the CMHA reported in the 18 to 24-year-old group a 60% increase in mental health issues. So that to me was really telling that, things are going on and there's not those coping mechanisms. So students from universities in Manitoba, all the way to the East Coast, that was a really interesting process for me, just getting REB approval at all of these different institutions. What was difficult though, was actually reaching the students. So, I mean, it took a lot of time for those REBs to be approved at, 8 to 12 universities in there. I can't remember the exact number giving us REB, but then actually communicating that this study was going on was the challenge. So we offered 8 weeks from May to July. Students could attend synchronously or asynchronously using the Padlet platform. And basically it was looking at, we had researched what are some of the more ancient wisdom principles that exist, and how can we focus each session around one of those principles. So like wisdom, joy, benevolence, honesty, those were things that we looked at. And I'll share with you in a moment. And so we invited people to join either way for 15 minutes a week. So it wasn't extensive. We just wanted a short 15-minute introduction to introduce the concept, invite them to reflect and answer some questions around that ancient wisdom concept, and then invited them in 2 separate groups, either to experience a 10-minute meditation or a guided writing activity. So before we started the study, we had 102 participants signed up and shared their informed consent. But when the study actually lifted off at the end of mid-May, we only had about 30-ish participants, and in the end, about 22 completing the program. So these are the different elements we looked at throughout the first 6 weeks. And then the last 4 weeks were focused on joy, acceptance, and then we held a focus group and we had some interviews. And for each of these concepts, we collected artifacts, as well as any writing that participants wanted to submit. So we have artifacts that we're analyzing right now and going through as well. So this is a mixed method action research study that incorporates a focus group interview. There was a pre-survey, a mid-survey, as well as a post-survey. And we also had the artifact submissions. So submissions could be written or any kind of artistic expression they wanted to share. So really this process, so what we found with the research in general from the past study is that in these kind of situations, participants notice more, they pay attention to details they might not have really thought about before. And it also broadens perspective and possibilities and has been shown to lead to creativity. I will also share that it can also trigger people. So it's important that we had, counselors and other people, hotlines that people could call, because this study was interesting in that I didn't really have any connection with the participants other than them watching me on a video and having some chat. So it was a really kind of inhumane disconnect where I didn't get to know them. And so having these counselors and numbers was really key to this kind of research. So this is something that I developed at the end of the first meditative intervention. And it was just a process of awakening led to focus. The relaxation led to greater empathy or self-care that led to either a notice of incongruence and a wish to be more congruent, that alignment between your values, thoughts, feelings, and behaviors, and seem to have a better confidence in your participants' ability to want to move forward, which led them to be a little more inclusive and creative. So I know there's a lot of detail on this side, but that's just the essence. So I'm going to pass this over to Haley so she could share some of the preliminary information that we have discovered and are inspired by.
Okay, perfect. Hi, everyone. So this is some of the I guess, data that we collected after everyone kind of went through each week. So this specific screen that's being shared is after our wisdom week. So the participants were given certain prompts after they did the teachings with Jody and other meditations, things like that. And this specific question that they were asked was to write a letter to your past self and just kind of let yourself know what wisdom would impact you on any previous version of yourself. So these are kind of just some answers that people stated through their submissions. So I'll let everyone take a moment to read a few of them. Yeah, and it's pretty interesting just seeing how some people's were very similar and then others were kind of different. But yeah, I think that's it's really up to everyone to kind of read and see how you interpret it as well, which is a really nice and beautiful thing about more qualitative data. Okay, and then the next one, these are some artworks that were submitted by participants, and this was another question during the Wisdom Week that was asked to kind of do a piece of artwork or even write about your energy as a child or your childhood self. So as you can see, these were two kind of very different pieces of art that were submitted. And it's just kind of lots to look at, I feel like, but it's kind of interesting to interpret. But I don't know, I love seeing everyone's artwork. I thought it was so amazing just to see what everyone would put. But yeah, I think that's most of it. Awesome. I'll hand it back to Jody. Thank you.
Thanks, Haley. And the comments here, Haley, I can't quite read them. Is it like anything but here? And it says the whole.
Yeah, it says anything but here, this whole will do.
Thank you. So that concludes basically the presentation, and hopefully this inspires, the people who are engaged in this research and myself and other people involved that a tiny, tiny change today brings a different tomorrow. So I have the references here and I'll stop sharing, but if there's any questions, I can answer those now.
I don't see any questions in the chat, and I don't see any in the Q&A, but I'll give everyone another minute to add some in. We do have a hard exit at 12:58, so that's in 2 minutes, just so the next group can log into the webinar. So I'll give it another 30 seconds in case anyone wants to make any comments or have any questions. Okay, so I don't see anything right now, but if anyone does have comments for Jody and Haley, feel free to send them along to their email at AwakenU, or connect with the Office of Research Services and we can pass that along. So thank you so much to you, Jody, and also to Haley for sharing AwakenU with us. I'd also like to thank Paulette and Bill for presenting their research with us today as well. It's always exciting to hear faculty's research and be able to share that with the Algoma U community. So this session ends our second annual research week. However, if you missed any of this week's presentations or haven't yet had a chance to visit the Research Week website, all content will be available year-round at auresearchweek.com. So thank you to all of this week's presenters for making this another successful research week. We look forward to planning next year's event, which will probably look a little bit different than this year's, as we can hopefully, fingers crossed, host live events on campus, on all 3 of our campuses. So have a great weekend, everyone, and thanks so much for joining.